Hello everyone, myself Dr. Pushpa, Associate Professor and HOD Microbiology Department of Ramaya College of Arts, Science and Commerce. Welcome you all to my video lecture on smut disease. The last video we had started with the study of fungal diseases, wherein which we had studied about rust disease. Today let us study about smut disease through this video. So, before going to the detail of the smut disease, why this disease has been named as smut disease? This is due to the presence of black powdery mass of the teleospores in the infected part of the plant. The infected part, usually the seeds or the ovaries of the flowers, or the infective region, wherein which you can see such kind of brownish to blackish powdery mass of the spores. The spores are called as teleospores. It is also called as smut spores. Once again I will repeat, smut. Well, the disease is called as smut because of the presence of black powdery mass of the spores in the infected part of the plant. Now, let us study about this disease in detail. Smut diseases can be classified as loose smuts and covered smut. What are these loose smut? As the name indicates, here the infected part, that is the ovaries, are the target region of the infection, wherein which you can see the entire ovary or the grain, or the ovary which has to produce the grain would have been accumulated with the black powdery mass of teleospores, and once it accumulates inside the ovary, it would have got covered over by a delicate membrane. Later this membrane ruptures and all the spores are disseminated and the spores are released. That is the reason it's called as loose smut. Whereas in case of covered smut, once inside the ovary, the smut spores or the teleospores are produced, they are not released. They are not released at all. Where each of the ear of the ovary of the smutted ear contain compact spore mass called as smut sorus. The grain is replaced by smut balls or sori. The spores are very compact that they do not fall apart. They are intact due to the deposition of fatty substance, and the spores not released or dispersed. They are held by a wall of the ovaries and the basal part of the glumes and the spikelets, and there the smut spores are also being covered over by a white shiny membrane and not blown out by the wind. That's the reason it's called as covered smut. Once again I will repeat why it's called as covered smut. Once the teleospores are produced inside the ovaries, these teleospores are not released at all. These teleospores are in the form of sori or in the form of sput balls. They are so compact to each other, and this compactness is due to the deposition of fatty substance, and these spores are not released or they are not dispersed. They are held over by the wall of the ovaries and also the basal part of the glumes and the spikelets, and the smut spores are being covered over by a white shiny membrane, delicate membrane, and they are not blown out by the wind. That is, they are intact along with the ovary or along inside the inflorescence. You can find them intact. They are not released at all. The spores are not released. Instead, they are covered over by a delicate membrane. That is the reason it's called as covered smut. That sori of the smut or the smut balls have been covered over by a delicate membrane and they are not disseminated through wind. That's the reason it's called as covered smut. Whereas the loose smuts, once they have been formed, once they have been accumulated inside the ovaries of ovaries of the flowers, they have been ruptured. It has been ruptured and they are disseminated in wind. That's the reason it's called as loose smut. Here in this particular slide, I have given some of the list of the important organism which causes the loose smut. Ustilago medis, which causes the spot disease in case of corn, where we are going to study in detail about this corn smut in my future videos. Ustilago avinae causes loose smut in case of oats. Ustilago nuda, Ustilago tritici, it causes smut in case of wheat. Important for the point of competitive examination. Ustilago nuda, Ustilago tritici, which causes the smut disease in case of wheat. Loose smut that you have to concentrate on. And you have Sphaerotheca sorghum. It is one of an organism which causes the smut disease in case of sorghum, and also Urocystis. Urocystis is one of the genus Urocystis cephulae is one of the genus or the species of Urocystis which causes smut in case of onions. This you would have seen, wherein which you can see some of the black spores in the onions. That is nothing but the Urocystis cephulae species which is been causing the smut disease in case of onions. And we have covered smut in case of barley and wheat, where in case of barley it is Ustilago hordi which causes the covered smut in case of barley, whereas Tilicia tritici which causes covered smut in case of wheat. So remember the species of the different genera which causes loose smuts and covered smut, and the difference between the loose smut and the covered smut has to be remembered. Now let us study about the loose smut of wheat. As I was mentioning before, loose smut wherein which once the smut spores are formed inside the ovaries, the wall of the ovary or the membrane which would have covered these spores ruptures, and all the spores, that is teleospores, or also called as smut spores, are released and they can be disseminated through wind. So that's the reason it's called as loose smut. So now let us study where actually you can find this particular disease. This is a disease where it is worldwide in distribution in humid as well as in semi-humid region. In India, we can observe this particular disease wherever the wheat has been grown. So today let us study about the pathogen Ustilago tritici, which is going to cause the loose smut in case of wheat. So let us study the systematic position of this particular organism. As we all know, this is an organism which belongs to the subdivision Basidiomycotina, class Teleomycetes, order Uridinales, family Uridinaceae, the genus Ustilago, the species Tritici. Coming to the disease symptom, this is one of a disease. It's a seed-borne disease, as I was mentioning in my previous videos. So the diseases can be airborne, seed-borne, soil-borne, etc. So this is one of a seed-borne disease where we can see the infecting pathogen or a propagule found on the surface or inside the seed. So this is one of a seed-borne disease where we can see the pathogen inside the seed. So if you see the symptom, no symptom can be observed until the plant has headed. What is the meaning of this headed? We cannot see any symptom till we can see the inflorescence. Inflorescence, till it forms the inflorescence, no symptoms can be observed. The infected plant as well as the healthy plant are similar. However, the diseased plant head earlier than that of a healthy plant. That is, the infected plant produce the inflorescence, or it produces the flower before the healthy plant blossom. And in some varieties, like in case of Sonalika, you can see the formation of yellow chlorotic streaks which may develop on the flag leaves. What are these flag leaves? In the left hand side of this particular slide, you can see some of the leaves which has shown yellow chlorotic streaks. What is the meaning of chlorotic? Chlorosis, that is the loss of the photosynthetic pigment, loss of chlorophyll, is called as chlorosis, wherein which this kind of symptom is being observed in some of the varieties where the flag leaf, that you have to remember, flag leaves become chlorotic. What is this flag leaf? The leaf which is found just below the panicle. That is, the leaf you can observe in this particular slide, in the left hand side down, you can see there are some leaves which are found just below the inflorescence. Those leaves are called as flag leaves. These flag leaves are very much important because they are also contributes in the supply of photosynthetic products to the panicle. So they are very important. Physiologically they are very important because they contribute in supplying the photosynthetic products to the panicle. Panicle is an important part of the plant. When it is concerned with that of a wheat grains are produced inside that particular panicle, where for the production of this wheat, the photosynthetic products which has to be supplied to the panicle, flag leaves plays a major role. So here in this particular disease, you could see the formation of yellow chlorotic streaks on the flag leaves. And also these flag leaves, the tip of the flag leaves become yellow, the tip becomes yellow, and also these flag leaves may also shed, that is they can just droop or it can just fall down after the process of chlorosis. They get dried and they can just shed. So that is the another symptom. In the infected plant, all the spikelets and the kernels in the sense they are grains, that is the wheat grain, and each of the head are smutted, and each kernel will be transformed into the smut mass containing teleospores. So here, see, if you see the left hand side of this particular slide, the first image which I have put. So these are the inflorescence, or these are the spikelets, and in each you can see the kernels, that is these you can also call this as a head, and in each head you can see some blackish color development, this wherein which these were the one which was supposed to get developed into the kernel, but it is not happening because the plant has got infected and the pathogen has entered into this inflorescence and even to the ovaries, and the ovaries which were supposed to produce the grain has been occupied by this particular pathogen, and there inside that particular ovary the spores would have developed. That is the reason each of the kernel in the sense you can call it as a grain, has been transformed into the black powdery mass of teleospores. Later, initially, each of this smutted kernel would have been covered over by a delicate membrane which burst, and all these powdery mass are released outside. They are released free and they can just disseminate through wind and it can infect the healthy plant. So once again, I will talk about the disease symptom. As I was mentioning, this is one of the seed-borne disease. You cannot differentiate the healthy as well as the infected plant initially. Later, what happens? The diseased plant start producing the inflorescence or the head before that of a healthy plant. Later, in some cases, in few varieties, you can see the chlorotic streaks on the flag leaves, which are very much important, um, which are found just below the head or the or the inflorescence, and these are very important. They produce the photosynthetic product. They supply the photosynthetic product to the panicle, so they get infected. You can see the chlorotic streaks over there, and if you see the inflorescence or the head, you can see the kernels, where in which inside this kernels you can see the entire tissue would have been occupied over by a teleospores, and the smutted kernels which have been covered over by a delicate membrane ruptures and releases the large amount of these smutted spores. Next, the disease cycle of this loose smut. So as you can see in this particular life cycle, towards your right side of this particular slide, you can see the inflorescence or the head, which or the kernels, which has been ruptured, and it has released large amount of teleospores. It has released large amount of teleospores. These teleospores, which are dispersed or blown away by air current. They are dispersed or disseminated by wind, and once it come in contact with that of a healthy plant, healthy inflorescence, healthy flower, so this is what it happened. So the flower of the wheat, it has got a the gynoecium has got three different part, which are those: style, stigma, and a ovary. Here the stigma is feathery stigma, and it has got a style and a ovary. The teleospores, which have been produced from the infected plant, once it gets disseminated, it falls on a feathery stigma. These teleospores start germinating. Initially, these teleospores are dikaryotic. dikaryotic. Later, these dikaryotic teleospores, they germinate, and produces the cells, where each of the cells are monokaryotic. That we have to remember. Once again, I will repeat: the teleospores, which are produced from the infected plant, once they are dispersed or disseminated through wind, once it come and land over on a feathery stigma of a gynoecium or a flower of the wheat plant, it germinate and produces dikaryotic hyphae. This dikaryotic hyphae, soon it come in contact with the opposite strain of the hyphae, that is, they undergo a process of karyogamy to form a dikaryotic cell. Soon after the dikaryotic cell has been formed, the dikaryotic cell start entering inside the ovary. There are many ways in which when the different mycologist has been interpreted. It can enter through the style, and they can enter into the ovary, like how the pollination and the fertilization pollination happen. In the same way it can enter. Or some of the mycologists also tell that these dikaryotic cell, they penetrate through the wall of the ovary. So the entry of this particular organism can through the stigma, style, and ovary, or directly they can penetrate through the wall of the ovary. In these two methods they can enter. Once they enter into the ovary, they enter into the ovarian tissue. That is, the ovary is full of nucellus and the endosperm. Endosperm is one of a reserved food material for the growing embryo. Endosperm and also the nucellus is rich in the food material. So the ovary, this particular fungi enters into the ovary, and later it also invades the embryo inside the ovary, or after the process of fertilization, the embryo would have been formed. These embryo would have been protected over by a delicate membrane called as scutellum. This particular pathogen, the dikaryotic hyphae or the dikaryotic cell, once it enters into the ovary, since there is large amount of nutrition inside the ovary, they grow and develop inside that particular ovary, utilizing the nucellus as well as the endosperm which is present in that particular ovary. Later it also infects the scutellum, as well as it infects the embryo which is present inside the ovary. Scutellum is nothing but just like a cotyledon which you can find in case of dicots. In case of monocot, it is called as scutellum. Scutellum is the one which protects the ovary in case of monokaryotes. So once the infection has been happened even to the embryo, if such infected seed has been sown by the farmers, if such seed, this is now the seed where inside the seed you can find the mycelium of this particular pathogen. So the such particular infected seed, once it start germinating, what happen? So once this particular infected wheat is being sown by the farmer, once it start germinating, this mycelium, which may be in the form of dormant, if once the grain has been dried and all, they gets activated. They start developing. This particular mycelium overwinters inside the embryo and infects the infected inside the infected cereals and kernels. They start growing, they start developing, the mycelia start growing and it develops inside this particular seed, and later at the same time the wheat seed also start producing seedling. It also start producing seedling. Once the seedling has been produced, this particular the pathogen which is present inside the seed enters into the shoot system as to shoot system of the infected seedling. So here once again I will repeat, the infected seed has been sown. Once it has been sown, the mycelium which is already there inside the seed, they also start germinating. At the same time, seed which contains infected embryo inside them, they also start developing into root system as well as shoot system. The seed where the pathogen which is present in the seed also enters into this particular embryo or the seedling, and as the seedling is developing, this mycelium also develop. They enter in between, they invades the tissue of the infected young seedling intracellular, that is, they can enter even inside the cell of the host. So once they grow and develop, we have to note down that they are being found concentrating mostly on the apical part of the plant, because from this apical part of the plant, that is at the tip of the particular plant, you can see the development of the inflorescence. So once the plant is growing, even this particular mycelium also grow along with the seedling, and once the plant start producing this inflorescence, they also enter inside the inflorescence. They also enters into the into the flowers, into the gynoecium part of the flowers, and inside the gynoecium is the female reproductive structure of the flower, where it enters into the gynoecium, that is the ovary. It utilizes all the nutrient which is present inside the ovary. At by that particular time, even the pollination would have happened. Actually, the seed kernel was supposed to be produced, but the entire ovary, inside this entire ovary, this particular hyphae start developing inter as well as intracellular, and utilizes all the nutrition which is present in that particular ovary. Once the nutrition is being utilized, the entire nutrition present in that particular ovary has been utilized by this particular organism. They get the each of the mycelium, the cells, they become teleospores. They start producing closed septa. The dikaryotic mycelium which is present inside the ovary start producing closed septa, and each of the septa become encircled, that is they become round, and each of those rounded cells are called as teleospores. So at that particular time, the entire kernel or the entire grain or the entire ovary would have been occupied by such kind of round or oval shaped spores called as teleospores. Each of the teleospore is dikaryotic in nature, and it is made up of two wall layer, exine and intine. Exine is spiny, and once these teleospores are being occupied by the entire ovary, which would have been covered over by the delicate membrane, the delicate membrane ruptures, and this teleospores, black sooty teleospores, are released outside, and such teleospores can again infect the flowers, and the cycle can continue. Once again, I will repeat, this is one of the seed-borne disease. Seed-borne disease. The infection, the primary infection to the plant happen by the teleospores. Teleospores, once they land on the feathery stigma of the flower, they germinate, produces mycelium. Initially, the mycelium is monokaryotic. Later, they come in contact with the opposite strain, undergo the process of karyogamy or somatogamy to form a dikaryotic cell. This dikaryotic cell infects the ovary. The infection can happen either through stigma style and can enter into the ovary, or some mycologists tell that the infection can happen directly by the penetration of this particular mycelium into through the wall of the ovaries. Once they reach the ovarian tissue, wherein which you can find the nucleus as well as the endosperm, they can infect, they can grow in that particular seed, and also it infects the embryo. With the developing embryo, it can infect, and also it can infect the scutellum. The scutellum, once again I will repeat, this is like a cotyledon of the dicot, or it is the membrane which actually protects the embryo. So infects both the embryo as well as the scutellum. And once such kind of grain has been sown, once this grain start germinating into the seedling, at the same time this particular pathogen which is present inside the seed also start germinating, infects the seedling, and inside the seedling, if you take a transverse section and observe it under the microscope, you can see the mycelium which are intracellular, and as the seedling is growing, they also start growing, where in that particular moment you cannot see, you cannot differentiate whether it is an infected plant or a healthy plant. So they just grow along with the seedling, and they try to grow more towards the apical part, because that is the region where actually the inflorescence grow, and they also infect the flag leaves also. And once the inflorescence have the head has been formed, they enters inside the ovary of the gynoecium. There they utilize all the nutrition which is present in the ovary, where the ovary is one of a region where you can find the rich nutrition. Once the nutrition is over, what they does, they start becoming teleospores. This teleospores is being occupied by the entire ovarian tissue. At the same time, mass of the teleospores, which has been covered over by the delicate membrane, ruptures. Once the ruptures, these teleospores are released into the surrounding and they get disseminated through wind, and once this fall on the suitable host, the cycle continue. So how to control this particular disease? Since this is one of the seed-borne disease, better to go for seed treatment. Seed treatment, that is, you are going to treat this infected seed with carboxin. It's also called as Vitavax or carbendazim. It's also called as Bevestin at the concentration 2.5 to 3 grams per kg of the seed, or we can also go for Raxil. It is being produced by Bayer. It's a company at 1.25% concentration, and it is effective like that of Bevestin and carboxin. Use of certified smut free seed. This is the best method of controlling this disease. Use of certified smut free seeds. And also the farmers can go for solar treatment, that is soaking the seed in water for 4 hours followed by drying in sun for 12 to 4 from afternoon 12 to 4 p.m. during April to June. That's the summer season. In that particular season, the temperature also will be very high. At that particular temperature, we can go for solar treatment, that is soaking the seed in water for 4 hours followed by drying the seed for between 12 to 4 p.m. during April to June. Other method is soaking the seed for 4 to 5 hours in water at normal temperature. Later, transfer it into hot water at 54 degrees Celsius for 10 minutes. This is another method. That is hot water. You can also call it as hot water treatment. The best method is use of disease resistant varieties. Some of the disease resistant varieties are NP 710, 718, 761, Vasipalli, 808, Bansi, 224, etc. These are some of the methods to control this loose smut. Thank you. Thank you for your patience to watch this particular video. Thank you once again.